Hello and welcome to Biology Baking. Today we're making a cell cake. To make the cake, I begin by placing my butter, sugar, and vanilla into a bowl of an electric stand mixer. Then, I beat them together until light and fluffy. I added the eggs 2 at a time, mixing in between until fully combined. Next, I alternated adding my dry ingredients and milk, beginning and ending with my dry. I poured my batter into 2 9-inch round pans lined with parchment paper until they were a little over halfway full, and smoothed them over with a spatula. I baked them off at 350 degrees for about an hour and 10 minutes, or until a toothpick inserted into the cakes came out clean. Placing the cakes on a Lazy Susan, I carefully leveled them and cut the caramelization off the sides of both cakes, as well as off the bottom of the second cake. I then brushed both cakes with simple syrup to lock in their moisture while I worked on them. Beginning with my second cake, I placed it onto a cake board and spread on the filling. Next, I placed my first cake on top so its smooth side was on top. I thinly iced the whole cake to give it a crumb coat and lock in any stray crumbs, and refrigerated it for half an hour to allow it to set. Once out of the fridge, I spread on a thick layer of frosting all around the cake and smoothed it flat with the bench scraper, filling in holes as I went with frosting and smoothing them back over. Getting this layer as smooth as possible will prevent our fondant from showing any flaws later on. The cell membrane is a thin but strong boundary between the cell and its environment, allowing some things in but not others. I began by making my cell membrane out of a dark purple fondant by rolling it out to the cake's circumference, or its measure all the way around, which I measured with a tape measure. Once long enough, mine was 30 inches, I carefully wrapped it around the cake and trimmed off the excess. To patch up cracks in its seam, I mixed some leftover fondant with water to make a paste and filled in the gaps. Once dry, the paste makes it nearly impossible to see the mistakes. For the cytoplasm, I began by making a base from light blue fondant and rolled it large enough to cover the top of the cake. I laid it on top and carefully trimmed around the edges. To make the cytoplasm, a jelly-like substance that fills the cell and holds its organelles, I tinted clear piping gel with blue food coloring and spread it over the base fondant. This is the nucleus. The nucleus contains a cell's DNA and controls all the cell's functions. It has its own membrane, the nuclear envelope, which is double-layered and has tiny pores to allow ribosomes to exit. It holds the genes that instruct how to make proteins. To make the nucleus, I rolled out a piece of bright pink fondant and cut out a circle using a bowl. I used a knife to clean up its edges. The nucleus contains a nucleolus, which makes the cell's ribosomes. To make the nucleolus, I crumbled up a small piece of cake that I made with the leftover cake batter. I added a small glob of frosting and kneaded them together into a soft dough. I rolled the dough into a ball, covered it with a piece of light purple fondant, and cut off the excess. This is the endoplasmic reticulum, or ER. It is made of thin interconnecting membranes. The ER has two parts, the rough and smooth ER. Its rough ER is covered in small ribosomes and produces protein. The smooth ER produces lipids, or fats. To make the rough ER, I rolled purple fondant into several short snakes and placed them around the nucleus. Once arranged, I used a fondant tool to make lines in the snakes to mimic folded membranes and connected some of them with lines. I did the same for the smooth ER, but I made it a bit smaller. To make some round membranes, I rolled out some fondant balls and used a ball tool to make an indent into them, to make them look like tubes. Once finished, I used a mixture of clear extract and purple food color to paint on the ribosomes on the rough ER. This next part is called the Golgi apparatus and processes, sorts, and delivers proteins. It can store them, transport them to other parts of the cell, or send them to the cell membrane to exit the cell. To make it, I rolled short snakes and arranged them by length, as shown. Then, I used a tool to mimic folds on the Golgi body, another name for the Golgi apparatus. I rolled out small pieces of fondant to make vesicles, which are small sacs that transport proteins. I used two of these to make round tubes by indenting them using a ball tool once again. These are the mitochondria, and they are much more than just the powerhouse of the cell. They make energy, called ATP, for the cell, and even have their own ribosomes and DNA. To make them, I used electric green and dark green fondant. I passed the dark green through a clay extruder to form DNA strands. I formed the electric green into several bean shapes and used piping gel to adhere some DNA onto each. These are lysosomes, round organelles that contain enzymes to break down old or damaged cell parts, as well as defend against bacteria. To make them, roll out some round shapes out of orange fondant. These are vacuoles, and they are liquid-filled sacs that a cell uses for storing things, such as food molecules, water, and enzymes. To make them, roll out small balls of light blue fondant and use a ball tool to make them hollow on the inside. These small round bits are called ribosomes. They are made in the nucleolus and can be found on the rough ER. They make proteins in cells by linking together amino acids. To make them, I made long strands of purple fondant with my clay extruder and cut them into small pieces, rolling them out to make each ribosome. Hopefully you paid attention to the checkpoint questions throughout the video, because now it's time for the quiz. Question 1. What is cytoplasm? Question 2. What organelle is contained inside the nucleus? Question 3. What's another name for the Golgi apparatus? Question 4. What can vacuoles store? Question 5. Where are ribosomes made?